To start the synapse, and this is looking at the electrical passage of impulses from neuron to neuron. The first objective we're going to begin with is to understand the differences between chemical and electrical synapses, okay? And although we're going to look at both, I just want to make it clear here that the emphasis will be on the chemical synapse, okay? We'll talk a little bit about electrical synapses, how they differ, but you'll be expected to know the details of the chemical synapse more so. So let's talk about what a synapse is. It's an area of functional connection between a neuron and any other cell that a neuron is signaling. So neurons can be signaling other neurons if they're in the central nervous system. This is the brain, the neurons of the brain, the spinal cord. So when one neuron synapses another, this is typically in the central nervous system. Or a neuron could synapse other types of cells as well, muscle cells, and this can be smooth muscle, cardiac muscle, skeletal muscle, and glands as well can be synapsed by other neurons. And when we synapse in the central nervous system, it's typically referred to as a ganglia. We'll talk about how this examples of this in the autonomic nervous system coming up in the next module, but this is usually referred to as a ganglia. Whereas when a neuron synapses another type of cell that is in the peripheral nervous system or out in the periphery of the body, the skeletal muscles, glands, etc., this is called a neuromuscular junction. Okay. And it can also sometimes be called a neuroeffector junction. We're going to talk about all those differences again coming up. Now, when we think about two neurons in series, we have a presynaptic neuron, which is the first neuron, and its axon terminal is usually coming into contact with another neuron, and we can refer to that as a postsynaptic neuron. And sometimes you'll hear the term preganglionic or postganglionic again if it's in the central nervous system, right? So one neuron synapsing another, and we have a pre and a post referencing that synapse. Now, there are different ways in which neurons can synapse each other. We can have the axon terminal connecting to the dendrites of the postsynaptic neuron. This is considered an axodendritic type of synapse. We can have an axon terminal synapsing onto the soma or the cell body, and this is considered an axosomatic type of synapse. And then we can have two axon terminals synapsing onto each other. Now, this example, we're going to come back to when we look at the presynaptic inhibition and facilitation towards the end of this chapter, this unit. So axoaxonic synapses are when two axon terminals are in communication or synapsing together. Lastly, the most common, the most abundant synapse in our body is the axodendritic, okay, axodendritic. And we can also sometimes see axosomatic as well. Now, we can also differentiate a synapse based upon whether it is electrical or chemical. So let's look at how those differences we can dictate those differences. So basically, an electrical synapse is when that impulse remains as electrical energy. So an action potential, for example, an action potential travels from one neuron or one cell to an adjacent cell, and that energy is maintained as electrical energy, and so that would be considered an electrical impulse. Okay, a chemical type of synapse means that the electrical energy that is the action potential must be converted to chemical release, right? The release of chemicals at the synaptic junction, and then that chemical can cross the synaptic cleft, bind to its receptor, and then that can be reconverted into an electrical impulse on the adjacent neuron. So the difference is, and you can probably already predict, an electrical impulse is going to be faster because the action potential is going to maintain its form, but just go from one cell to the other. And with a chemical synapse, it's going to take a little bit longer time because the action potential arrives at the axon terminal, and then we have to convert that electrical energy into chemical energy, release those chemicals, and then that can be reconverted to electrical energy on the second neuron. So this is going to take a longer time just because of the steps that are involved. All right. But as I mentioned before, the chemical synapse is the most important, it's the most abundant, and it's the one that we're going to have emphasis on in terms of the depth that you need to know. So let's begin by thinking about the electrical synapse first. So two neurons are connected to each other by gap junctions. This can be between neurons and glial cells, and we'll kind of, talk about what those are in the next chapter. But glial cells are non-neuronal supporting cells. They are not electrical in terms of being able to initiate an action potential, but they can receive one from the neurons. Okay. Now, an electrical impulse is going to be rapid in its communication, which sort of makes sense. If the impulse is going right through the gap junctions, then there's no transference of this impulse. It's going right through faster, rapid communication. We can sometimes have ions or second messengers actually passed through those pores as well, those gap junctions. So for example, sodium can rush from one cell to the other, and that can be the way that the impulse transmits across that gap junction. These impulses are usually bidirectional, so they can, again, move from one cell to the other in both directions. And that is because there's no need to transfer this to any other form. It's going to remain as an electrical impulse and it can go right through in both directions. We're going to be able to see excitation and inhibition at the exact same synapse. So because it is a rapid transmission of the impulse, we can see both excitation and inhibition being signaled through these gap junctions. Now, again, it's very rare, but some of the examples of where we find these types of impulses in the body are the retina. And this is what helps us to transmit the light impulses coming in into action potentials, electrical energy that can be transmitted very quickly through the visual pathway. We also have the cortex and brainstem, especially the parts of the brainstem that are involved in breathing and the control of respiration. And then lastly, we have the hypothalamus and some of the neuroendocrine neurons that are projecting from the hypothalamus to other areas of the brain, especially the pituitary gland. Okay, so these are some of the specific examples, really only in the CNS, as you can see, where we have electrical impulses, right, between neuron and neuron. Let's shift and think about the chemical synapses. So chemical synapses, again, we usually have two neurons connecting to each other or signaling to each other, a presynaptic and a postsynaptic. The distance of the synaptic cleft here is going to be greater. Whereas in the electrical synapse, we have the two neurons basically tethered to each other and the gap junction being the passageway for that impulse. With a chemical synapse, it's really just two parts of the neuron, not necessarily tethered, but coming in close contact. And the distance here is about 30 to 50 nanometers wide. Now, the impulse here is unidirectional, so it's going to only travel in one direction. And we can see different types as we discussed earlier. So we can see an axodendritic, axon terminal to dendrites. We can see an axosomatic, axon terminal to soma, or the cell body. And then we can see axoaxonic, axon terminal to another axon terminal. But the most common will be this form, which is the axon terminal to the dendrites. Much more rare are dendrodendritic, so one dendrite signaling another dendrite, much, much rarer than even the other forms. Let's look at lecture objective number 2 to describe the communication across chemical synapses, specifically explaining how neurotransmitters are released and describe their actions after that release. So we can already begin to see here that neurotransmitters are involved. So we're not just dealing with electrical impulses here. We're going to convert that electrical impulse into a chemical release of neurotransmitters. And that is how the signal crosses that larger synaptic junction or that synaptic cleft. Now, before we get into the details of a chemical synapse, we want to think about the anatomy, the structures involved, the players involved. So we have our presynaptic neuron. which the axon terminal is where we're kind of focusing in on. And then we have a postsynaptic neuron, and we're focusing in on the soma or the plasma membrane of that. It could be the dendrites, which is the most common, or even the soma on the other side. We want to think about the neurotransmitters within the vesicles inside the axon terminal. So inside the presynaptic neuron, we have these tiny vesicles that have within them the neurotransmitter that is specific to the receptor on the adjacent cell surface. So the neurotransmitter here, usually acetylcholine, but it can be others. Acetylcholine is the most abundant, and we'll talk about that again coming up. So we have the neurotransmitter containing vesicles. We also have voltage-gated calcium channels. So when we talked about the different types of receptors and channels that we find along the membrane, concentrated on the axon terminal are voltage-gated calcium channels, which means that they are going to be sensitive to voltage. And when the action potential arrives at the axon terminal, they're going to open up in response to a specific voltage threshold. Then we have the synaptic cleft itself, which is the distance basically between these two surfaces. This is also called a synaptic junction as well. And again, it's wider than what we see in the electrical synapse. And then we have other players that are on the postsynaptic cell surface. So we have receptors that are specific to the neurotransmitter that is being released. We have enzymes, and these enzymes are usually responsible for degrading any excess neurotransmitter that is left in the synaptic cleft after the impulse has been transmitted. Lastly, we have reuptake molecules. So reuptake molecules are actually on the presynaptic cell surface, and they have a similar role to recycle and reuptake any excess neurotransmitter as well. Okay, and I just want to emphasize that a little bit, but we'll talk about it some more coming up. The clearing of the synaptic cleft is extremely important in ceasing the chemical impulse. When we're transferring that impulse from one cell to the other, as long as that neurotransmitter remains in the junction, we're going to have a continual firing of that impulse. So in order to stop the signal, we have to clear the junction, and that is what's going to allow the muscle to relax, for example, right, in between contractions. Okay, let's go ahead and illustrate some of this and actually look at the steps involved. Okay. If you have any questions in the meantime, please feel free to share those, but let's look at the steps here in a bit more detail. Okay, so we're looking at the synapse. Especially the chemical synapse. Okay, that's what we're focusing on here, as opposed to the electrical. So we want to think about the cell surfaces involved. So we have our axon terminal, and then we have our adjacent, let's say dendrite, for example, because that's the most common type of synapse. So we've got our axon, our presynaptic neuron, presynaptic axon terminal, and our postsynaptic dendrite, right? The dendrite, or the cell surface of that dendrite. We also want to think about the structures that are involved. So we know that along the axon, we have our voltage-gated sodium channels. We have our myelin in some cases. Okay, so we've got these nodes of Ranvier that we talked about. And then in between those, we have the voltage-gated sodium channels. Okay. So at these junctions, we have our voltage-gated sodium channels. So we can already begin to see the action potential propagating down the length of the axon. So that's kind of where this begins. And we talk about the details of the action potential, how it propagates down the axon, but we're going to focus in on what happens when it gets to the axon terminal. So that would be the first step, is the action potential propagates right down the length of the neuron, and then it arrives at the axon terminal. Now, when it arrives here, there are voltage-gated calcium channels on the axon terminal membrane. So let's draw those in. Here's our voltage-gated calcium channel. And in response to the action potential arriving at the axon terminal, we're going to see calcium rush into the cell, right? That is the ECDF for calcium. Calcium will rush into the axon terminal, and that is actually going to excite the axon terminal membrane, similar as what we see with an action potential, right? Just increase the membrane potential. And in doing so, that is going to trigger the release of the vesicles that are containing the neurotransmitter. So let's draw those vesicles in, right? The neurotransmitter containing vesicles that are basically storing this neurotransmitter inside this terminal, right? And we can just draw in these neurotransmitters. Okay. So as the calcium rushes in, we excite the axon terminal membrane. That is actually triggering the exocytosis of these vesicles. So these vesicles are going to get close to the membrane. They're going to dock at the membrane, and then they're basically going to dump this neurotransmitter out into the cleft. Okay, so we can label this region here as the synaptic cleft. Okay. Right. Now, we want to think about how the neurotransmitter is going to bind to its specific receptor. So we're going to put the receptor here on the adjacent cell surface. And as you can see, it's specific for the neurotransmitter in question. So it's going to be a specific fit for whatever neurotransmitter. Now, as I mentioned, this is usually acetylcholine, but there can be other types, such as norepinephrine and epinephrine, which we'll talk about. But that neurotransmitter binds to this receptor. And this is going to bring about a cellular response on the adjacent neuron. Now, we want to think about the cellular response as being fast or slow, depending on the type of neuron. So we're going to just put here for now, we're going to put brings about a cellular response. And that can be fast if this receptor is an ionotropic receptor, or it can be slow if this receptor is a metabotropic receptor. And these terms will become a lot more clear as we continue on. But a fast response means that it's going to be an immediate transference of this chemical neurotransmitter to an electrical impulse. A slow response means that we have few other steps in the way before we can reconvert this chemical into an electrical signal on the cell. So we'll talk about the differences here, but that will be determined based upon exactly what type of receptor, and we can see both. All right. Now, after that signal has been transferred by way of the chemical release, we want to clear the junction of any excess neurotransmitter. So we talked about the enzyme that is usually on the postsynaptic cell as the degrading, right, as the enzyme that would degrade any excess neurotransmitter. So let's draw that in as well. So here's our enzyme. Okay. And it too is a specific fit for the neurotransmitter in question. Now, this enzyme is usually called acetylcholinesterase. If the neurotransmitter is acetylcholine, then the enzyme here is acetylcholinesterase, right? Acetylcholinesterase. And this suffix here, ase, means that it can degrade or cleave or cut. And that is what it does to acetylcholine. So acetylcholinesterase is going to break down acetylcholine, and that is going to help clear it from the junction. The other option for clearing the junction is to reuptake or recycle it on the adjacent cell surface, right? So back on the presynaptic side. So we have another type of reuptake molecule on the presynaptic side, and that can simply reuptake the neurotransmitter, repackage it into a new vesicle, and that can be another way to clear the junction. So this is a reuptake molecule. Okay, and this is going to simply reuptake this neurotransmitter and repackage it into a new vesicle. Okay. Now, I did want to just list out the process here. We talked about exocytosis in membranes part 3, but this is also an example of exocytosis when these vesicles dump their contents out into the cellular junction, into the synaptic cleft. This process is called exocytosis. Okay, as opposed to endocytosis, which we also talked about, which is when we uptake those neurotransmitters into vesicles. Okay, but for this example, it's exocytosis.